Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. If you've ever wanted to measure ligand bias, you most likely have looked at the activation of different signaling pathways. But how does actually the binding of your ligand at the extracellular pocket of your receptor redirect the residues within the receptor in order to activate it? And that remains difficult to trace nowadays. My guest today is Anita Niveda, a computational biologist studying allosteric communication in GPCRs. She uses molecular dynamics and network analysis to map out signaling pathways to receptors. During her postdoctoral work on GPCR structure and function, one problem stood out to Anita. Can simulations quantify ligand bias inside receptor structures? One evening, running analyses alone in the lab, a pattern appeared. Communication routes towards G proteins differed from those towards arrestins. When she counted those pathways, the numbers tracked experimental bias. A computational framework for predicting ligand bias began to emerge. Those calculations she made linked microscopic communication to macroscopic signaling outcomes. Suddenly, simulations were not just visualizations anymore, but measurements, and measurement meant predictions for drug discovery. Before we dive into this episode, I invite you to check out the relevant links in the show notes. And now, let's dive into this conversation with Dr. Niveda.
Hello, everyone. This is Yamina from Dr. GPCR, and I'm very excited today because this has been a long time in the works to have with me Dr. Anita Niveda. I told you I'm going to mess up the name. It's a Saturday morning. It's been a long week, but Anita, I'm so excited to have you on today.
Hi, Yamina. Thank you so much for having me, and I'm really excited for our conversation today.
All right, so let's start at the beginning. Would you please introduce yourself, tell our audience who you are and what it is that you do?
Absolutely. Yeah, as you said, my name is Anita. I'm currently a senior scientist at a BioTech in Toronto, Canada. Previously, I worked in another company in Montreal where we were offering computer-aided drug discovery services. I was there. That was my first industry role, and prior to that, I was in California doing my postdoc in the lab of Dr. Nagarajan Vaidehi at City of Hope in sunny LA area. And that's the lab where I got introduced to GPCRs. And my PhD prior to that was in Georgia in the lab of Dr. Robert Woods. And my PhD was on working with carbohydrate molecules as ligands. And it was mostly focusing on docking and docking scoring functions, improving that for carbohydrates as ligands. That's kind of my grad school and current career situation. I moved to the US from India, actually, before that. I did my undergrad in India at Vellore Institute of Technology. I did my undergrad in bioinformatics. And that's kind of where my interest in using computational methods to study biological information started.
Wonderful. And we did have Dr. Nagarajan on the podcast, and I love chatting with her. She's wonderful.
Yeah, I actually listened to that podcast. I was very excited to see her on your podcast.
She is wonderful. Thank you so much for that introduction. I have to ask, how did you get interested in science in general?
Yeah, so I think science was it was something that I was always interested since middle school. I was always interested in biology, just studying anatomy. And there was a time I was interested in becoming a doctor, like a medical doctor. But I think that was when I was much younger, like I said, middle school. And then as I grew up and learned more about things like biotech and the possibilities that existed, my interests diversified as well. And I think around high school is when I got introduced to programming. I started programming. I started taking classes studying C and C++ programming, and I really loved it. And when it came time to, I guess, choose my major for undergrad, I wanted to choose something that combined both of those things, but I didn't actually know that those kind of fields existed at the time. And so that's And then when I came across bioinformatics, I was like, this is it. Wow, this kind of combines both of the things I like. And so that's so my interest in science has always been there. And then also with computers. And so that's what I still do.
I love it. And it's, I think other than you and Vaidehi, I don't remember ever having female computational scientists on the podcast. And that is, today is International Women's Day. So we're going to kind of reveal when this podcast is being recorded. And I was wondering if you can speak to your experience being a computational scientist and how, what's the ratio in your job of female versus male computational scientists?
Yeah, that's a good point. I noticed that we're recording on Women's Day as well. And I think when you spoke about me and Vaidehi being the only female computational scientist you've had on the podcast, I'm a little surprised, but I'm also not too surprised because over the course of my career, I guess starting with, I think my PhD lab was more, I think there was a balance of female and other scientists as well. But it was not just a computational lab, it was also experimental. But I would say since then on, there's definitely been, I've definitely been very outnumbered in a lab. Like when I joined Vaidehi's lab, I think I was apart from herself, I was the only female at the time. And then we had more women join the group later on. And I would say that's been true since then as well. So it's always been, I would say, underrepresented.
That's unfortunate, but I have you here today. And so if you know of any other female computational scientists in the GPCR field, please send them our way because we really want to highlight everyone in the field. And that's one thing that we're very passionate about, no matter what levels you're at, no matter where you're working, if you're working on GPCRs, on the wet lab or dry lab. But I think our focus here today is about the dry lab and the computational chemistry work. We'd love to have more guests.
Oh, absolutely. I will have a list for you.
Thank you so much. So you mentioned that you joined the Nagarajan lab, and that's when you got first exposed to GPCRs. And I was wondering, do you remember when you first you looked at a GPCR on a computer? Was that just another target, or was it something that you thought, oh wow, actually I'm seeing something that I'm really interested in, I could continue working on it?
I'm trying to recall when I first saw it. I mean, it was definitely probably in VMD or something, one of the programs we commonly use. I think even at the time when I joined the lab, I didn't kind of realize the wonderful world of GPCRs as much as I do now, about how much there is to study and how interesting they are and how diverse and how popular they are as drug targets, so just even in academia to study their biology. Because prior to that, so my PhD, as I mentioned before, I worked with carbohydrate ligands in a lab. The lab was focused on carbohydrates. And so the focus was on the carbohydrates or the glycans interacting with proteins. So proteins were the thing that was interacting with the main thing we were studying. And so when I was applying for my postdocs, I was specifically interested in just studying proteins in general. I just wanted to study more about proteins, more about their structure, function, their dynamics. And so that's the kind of labs I applied to. And I wasn't specifically looking for GPCR at the time. And so I think just having the opportunity to then be in this lab, where I could start working with proteins, I was super excited to get that role. But I think I started appreciating GPCRs more and more over the course of all these years, really. It's just grown because I think it's just such a great field to be in and to contribute to. And you just keep learning more and more, and people keep doing awesome research and publishing work. And I'm very grateful to be in this field.
That is wonderful. Can we go back a little bit to the time when you mentioned that you were applying for postdoc labs? So a lot of people who listen to the podcast are in those phases of, oh, I'm looking for a PhD lab, I'm looking for a postdoc lab. In your experience, how did you go about it, basically? And what if you could go back in time and tell Anita looking for a postdoc lab, what would be your advice to her?
Yeah, that's a good question. It seems like so long ago, but
not.
Sorry.
I said so long ago, but not.
Yeah, but I guess it seems like so much has happened since then. But no, it was such an important decision, but it was also a very stressful time because I was in the US and I was on a student visa, and I basically had like 3 months after my PhD to find my next role. That's the requirements. Those are the requirements of the visa. And so I think I defended in December, and I found this role in like January or February. But before I graduated, I started applying for jobs. Even at that time, I think I wasn't sure if I would go into academia industry. So I applied to jobs in industry as well as academia. It wasn't very clear to me what I should do. And I also needed to find a job. It was not, I was also like, I guess I couldn't be too picky about it. I intended to stay in the US, and I intended to work in science and to continue in the same field. And so I applied to a lot of jobs. I had a spreadsheet where I maintained all the places I applied to, when I applied to them. Like, it was a whole thing. But like I said, with postdoc labs, I did try to find labs where I was, where I thought I could expand my skill set and not do the same thing I was doing during my PhD. I consciously wanted to do that, to just learn something more. And I thought a postdoc would be a good environment to do that. The funny thing with my application to Vaidehi's lab was she didn't have an open position at the time. Like I said, I was doing everything possible to try to connect with people and try to find out if they have openings in the lab. So I think she had a very expired job ad for a research associate from 2 or 3 years ago that I came across, and that's how I got introduced to such a lab exists. And then when I went and checked out the website, I was like, this sounds like really cool research and I would love to be a part of it. So I think emailed her and I was like, I'm looking for a role. And then she was interested in my profile, and we had interviews, and then the rest is history. So that's how I got my postdoc role. I mean, my time at the lab has been such a great learning experience. It was a really good environment for learning. But yeah, I mean, you asked about how I got my postdoc, and
I love it. There's so much value in what you just said.
Okay.
And thank you for being so candid. And by value, I mean that you you just told everybody what to do when you're looking for the next one. For example, you had a spreadsheet where you were tracking where you were applying and when you applied, because I think that is important. Anyone who knows me enough knows that I like to track things. I like to know when and what did I do when and how long it took, because sometimes you apply for a job and you don't hear back for 30 days. And just because you apply for one job doesn't mean that you're going to get it. So you need to apply for multiple jobs. And also you have to remember to track those in order not to apply twice for the same job.
Exactly. And exactly, that was one of the intentions. That's an important thing, to not apply to the same job twice, but also sometimes if it's an email you sent to follow up, say in a week or something like that. So I found that very valuable.
Yeah, no, absolutely. I think the follow-up, again, is a great point. Just because you sent out one email one time, that doesn't mean that the person is going to respond, not because they don't want to. Maybe they didn't even see your email.
Yeah, absolutely. Those things can happen. I mean, I'm sure professors get a lot of emails from interested candidates. So yeah, you do your part and try to not be too pushy, obviously, but yeah, the spreadsheet really helped me at the time, and I still have it.
That's wonderful. The other point that you made that I really, really liked is the fact that you were trying to talk to as many people as possible and network.
Yeah.
In order to know who's doing what. And I think that also informed your decision as to, okay, what you wanted to do and what you didn't want to do, which is also very important.
Yeah, absolutely. I think the networking was a big part of it. Even while I was applying for jobs, I think that was part of my networking. It ended up being part of my networking. I got some good connections at the time with people in the industry who've been kind of like mentors. Like I actually there was someone I spoke to who was in a startup at the time, and it was a woman in science, and she was so gracious in they didn't have any roles for me, but then she was really encouraging, and she took the time to write me back an email and give me advice and just encouragement, and that was such a huge deal at the time for me, like looking for jobs in a relatively new country. And then we even met up and we had dinner or something. We met up for a meal, and I've been able to keep in touch with her with things that have been going on, and yeah, so those kind of things really touched my heart, especially when people responded to me. Even if they didn't have something, they would respond, and just if they had the time to do that, that was really great.
I think that's wonderful, and that's a great story. You never know who you might meet. Maybe you're gonna meet, your next best friend or a good mentor, or somebody who can say, "Wait a minute, we're not hiring now, but I know I have a friend who's hiring at their company."
Yeah.
And it seems like you entered these, conversations without necessarily demanding anything. You just wanted to get more information.
Yeah, exactly. Like you said, the possibilities are endless. You can never imagine what's possible by talking to someone. They can give you information that you never knew about job applications, for example. And so it's definitely a very valuable thing to do at that, just networking in general.
Wonderful. Let's go back a little bit to your PhD, and actually mainly your postdoc, considering that you worked on GPCRs during your postdoc. I'd be curious to hear more about what were some of the questions you were trying to answer during your postdoc, and what were you able to answer in the end?
Yeah, definitely. So I think, my first, the main, so as I said, I was looking to work with proteins and I got into this lab. The bonus was I got to work with GPCRs. And the other bonus was I also got to dive into the world of allosteric communication and proteins, which Vaidehi and her lab had been working on prior to me joining. And they had developed a software to study allosteric communication in proteins. Allosteric communication is basically within GPCRs. You can think of how the ligand binds to a GPCR, and the result of the ligand binding is you have G protein activation, beta arrestin recruitment. How does the ligand binding on one end, in the extracellular end, may affect these changes in the intracellular end? That's through communication through the receptor, and that's allosteric communication. You know about allosteric modulators and things like that. So I got to really tap into that and understand. It helped me understand how proteins in general work, like structurally, the dynamics, and how signal transduction could happen within proteins. And the main thing, I guess I did a couple of things while I was in the lab. One of the things was to understand ligand bias and how we can use computational methods to study ligand bias in GPCRs. So I used this tool, and I did some coding to kind of extract information from molecular dynamics simulations to quantify ligand bias computationally. So we applied that to several GPCRs with known experimental bias values, and we saw that there was a good correlation between the computational and experimental values. And this was actually a project in collaboration with Boehringer Ingelheim in Germany, and so I got to lead that project, which was such an incredible opportunity for me. And to actually be able to get something good out of it and be able to publish it was really, it opened a lot of doors for me during my postdoc. It got me a lot of invitations to speak at conferences, present my work, or even do. I had the opportunity to present that work as a poster in various places. And even within my organization, I was able to get recognition for that work, and so that was really, I don't know, that was a very important point in my career, I would say. And then I also then extended the allosteric communication to study subtype selectivity within GPCRs. So I looked at various subtypes of different GPCRs and how certain ligands can have different affinities to different GPCRs, and how can allosteric communication help us understand those differences. So that was something else I worked on. I got to work a bit more with doing collaborative work on angiotensin receptor, which is like a peptide GPCR. So that work kind of continued on even after I left the lab.
That is wonderful. And I think, and to your point, it is very important to understand bias also at a molecular level, not only to see which pathway gets activated, because it allows you to potentially, at least it has the potential to get a map of what happens. What are the residues that are interacting if you're activating G protein versus beta arrestin? And also receptor selectivity, subtype selectivity is also very important. It's a problem in drug discovery, not only in just general science, especially with some of these families of receptors that have different subtypes, and they all look similar, and it makes you wonder how do the cells differentiate and figure out, okay, now I need to activate receptor, I don't know, D1 versus D2, or this type versus other one.
Yeah, exactly. Yeah, what you said, I think you understood it well, what I mentioned. So there are these communication pathways that connect, for example, your ligand binding site to G protein or beta arrestin, and that's comprised of all these amino acids along the way. So it also gives you an insight into which of those amino acids are most important, and maybe you can target those for whatever, using an allosteric modulator, for example. So yeah, I found that it has applications in so many different it can be used to study GPCRs in different ways. And yeah, I was very grateful to be able to learn that in the lab and apply it to my work.
That's wonderful. That's wonderful. And I think also understanding what does that ligand binding pocket look like, and where does that ligand bind? What space does it occupy? I know water is important, water networks in there as well. So being able to kind of at a molecular level understand what are these interactions that are important, not only for ligand binding, but for activation is absolutely crucial. And you can't do that by running a cyclic AMP or a beta arrestin assay. You can't do that by just getting one snapshot with the cryo-EM structure. You need to be able to model these.
Yeah, absolutely. I think by being able to do molecular dynamics simulations, you can simulate proteins in motion and try to understand the different changes, conformational changes that happen. And with the subtype selectivity project, I remember what was cool was we knew there was a ligand which was more which had a better affinity for one of the muscarinic receptor subtypes versus the other. Doing MD simulations, I actually saw that the binding pocket was smaller, like it came closer to the ligand in the one with the better affinity compared to the other one. So it was really cool to see that, to have some validation or understanding of how the differences in affinity might look.
At a ligand receptor interacts, like one receptor hugs the ligand more tight than the other one, and the consequences. That's wonderful. You mentioned also that you had a really great experience working on the first project, which was in collaboration with Boehringer Ingelheim. And I want to get a little bit deeper into some of the things that you learned through this collaboration, because this was academic industry. And what didn't you know before going into, and what did you learn after from this collaboration? Not necessarily only scientifically, but also, as a scientist who then moved on into her career to work into industry.
Yeah, that's a good question. I think my collaboration with Boehringer definitely influenced. Is part of what influenced my decision to after my postdoc to apply to industry jobs. It gave me. We collaborated with like our main collaborator was a computational person within Boehringer who had like a lot of GPCR background. So it was a really cool collaboration because this was a collaboration where the collaborator brought a lot of knowledge to the table as well. A lot of, I would say, like the suggestions and ideas that came from the collaborator was directly very relevant to the things I was doing. So they were great ideas. And the person itself was a very approachable person and just easy to work with. And that was my first introduction to industry, and it was a very positive experience. And what I learned, I think, at that collaboration is also kind of how important GPCRs are to the industry. Like I think once when I started in the lab, it was almost like in a few months I started on this project. So I was just learning that GPCRs are important drug targets, but to actually see the interest from industry and see how they might use this for other projects, this method that we've developed was really encouraging. Like I remember thinking to myself, so they're going to use my method to do things, to work on other receptors that probably they can't reveal to me what they're working on, but it's probably something really cool and it could make an impact. And so that was very encouraging to me to be doing that. And I also got to work on more projects with them in the following years. And so just it was a very positive influence. And I learned that sometimes the research in academia and industry, you think about it as being very, like it can be different. But in this case, because we were collaborating on this project, I think I learned how it can be really fun and interesting to work in industry too, to work on these kind of problems. I think overall a very positive experience.
That is wonderful. And to your point, I think academia and industry are two sides of the same coin. And sometimes industry doesn't have the time to ask the very specific, very pointy questions about what does this residue do and this receptor. That academia has the opportunity to do that. And I think one feeds into the other, and the exchanges can be very valuable.
Yeah, I 100% agree.
Wonderful. So after this experience and you were finishing up your postdoc, I guess you just mentioned that, the collaboration with Boehringer was one motivating factor for you to go into industry. How did you go about looking for your next step after your postdoc? I guess I'm curious to hear anything that went well, anything that looking back now, you would have done differently. And this is all to help inspire our listeners who are in the same situation. We've all been there.
Yeah, I know. I completely agree. So yeah, I think after my postdoc, I wanted to explore more roles in industry, and I started applying for roles that seemed like a good match based on my expertise, and so I was looking at the usual places, I guess, the different job websites, but also just going into companies' websites and looking for roles there, because not all the companies advertise in common platforms. So that's kind of how I applied. Again, I applied for many companies at the time as well. But I would say actually that at the time, when I say many, for the kind of role I was looking for, there weren't as many openings as I feel opened up after the time I started. I think I managed to kind of keep track of job openings and stuff even after I got my role in industry. And I felt like there were more and more openings coming up later on. But for the specific kind of work I was looking for, there weren't as many openings. So I pretty much applied to everything that I saw if it was a good match. So that's what I meant. And I'm trying to think of what all I did. Sometimes if it was a LinkedIn job with a job poster, then I might reach out to them as well to send a message like, hey, I'm interested in this role, and just to help them maybe pay attention to my application a little bit more. Networking, I was doing a lot of networking, going to events. There were, I think it was called Association of Women in Science. I was a member of AWIS in the LA area, and I used to attend their events. And it was really cool. I just got reminded from your question, but they once had an event at a winery, and it was it was such a nice environment to meet other women in science, and it was a mix of women in academia as well as industry, and I got to network with people there and kind of learn about their different roles. And the women were not just they were not people just in research. They were also like I would say, what do you call these kind of roles? Like in regulatory affairs or more senior management. So like a whole range of perspectives, and it did open my eyes to all the possibilities in industry. So that was definitely a plus, like something I highly recommend to do. Like even if you don't know what will come out of it, just be there, just show up, and you even if you make one connection, that's worth going. And
Go alone to these events? Because I know it might be intimidating, especially when you're in academia and you're more of the junior side.
yeah.
I'm just curious to see if you went alone or not. I can already tell you that I did go to some of these events. There was a lot of women in bio events and things like that, and I always, found a friend to go with just in case, in case you were. But that's also dangerous, because if you go with someone you already know, then you have the tendency to talk to them. So what we did with my friend is that we would spend 10 minutes together when we got to the event, and then we would split up for 10 minutes, and then we'll make sure that we mingled with everybody. But it can be awkward, right? When you're going, you have 2-3 people talking at this types of events, and you feel like, oh, can I just jump in and say hi, or what do I do? And it takes practice, and at some point you get to a point where you figure out what are the best ways for you to network. And not necessarily go to these events, but find other ways to do it. And there are other ways to do it. I'm curious, did you go alone?
Yeah, so I think most of the time I went alone, just because. So when I say these, so these kind of events, but I'm also thinking of things like conferences where I had the opportunity to network. It was a mix. I did go alone several times because maybe I would be the only person from my lab attending a conference, and in that case, I'm by myself for like 5 days, and so I network on my own. But there were times I went with people from my lab, or I met someone there that I already knew. But I think most of the time we would still split up and try to like network, but also have like a familiar face to go and talk to whenever you need it. And I totally understand the need for that, because believe me, I felt awkward about just being there and just trying to network with new people. But the best part for me about conferences and these kind of events is having this topic or knowing that you can talk about science, you can talk about work, and you always have that subject to talk about, and talk about like what you're doing. And so it's not as open-ended as meeting someone outside in a social gathering. You do know there's something you can talk about, learn more about the other person. I think that's a great way to have a conversation. You're right about, if you go with someone you really know, it's great that you and your friend have that understanding to split, to the value in like splitting up and trying to network, because sometimes that can be a downside of maybe going with someone is to maybe you try to hang around with that person and miss out on opportunities to network. So as long as you have that understanding that it's important to talk to new people, then I think that's a great setup, to be honest.
Wonderful. And I also remember, so this person I'm thinking about here, we also practiced when we were both looking for jobs in industry. We also practiced interview questions. When we did that over the phone, and we like picked, okay, today we're gonna, obviously we're not in the same field, so these were not technical questions, but these were more behavioral questions. So we spent 20 minutes asking each other. It was always one person at a time, one call, one person being the interviewer and the other one being the interviewee. And at the end of that, we would give feedback in order to kind of, because you need to be able to try it. It's like running a pre-plate when you're running a cyclocam piazza. You want to see what that looks like. It's just like, and you can make mistakes there. And by practicing, you reduce the chances of saying some things that are not necessarily specific to the question that you're asked during an interview, because your stress level is, no matter how cool you are, your stress level always is just a little bit higher whenever, you're actually interviewing.
Oh, yeah, absolutely. Actually, that sounds like you had an amazing friend that you could do all these things with, where you had phone calls practicing for your interviews. That's really cool that you did that. Was this like when you were doing your postdoc?
Yes, it was during my second postdoc where I decided I was no longer wanting to work in academia, and we started figuring out what are the different networking events we might go to, what are the types of jobs we were each looking for. We're coming from two completely different areas. And so, yeah, we found networking events where it wasn't specific to one field. And after that, we did a lot of research online, and we actually ended up being supporting each other on this path. And every time one of us would have an interview, we would like spend a couple of days before doing these phone calls and giving each other feedback. And and we were very serious about it because we were really in our roles, of interview. And it was awkward the first time. I have to say, it was awkward the first time when I was the interviewee. And she would be very like, okay, can you please tell me about a time or something like that? And I started laughing and I said, okay, let me get back to to being serious. You're the interviewer.
That's a really great idea because, as you said, interviews are stressful. And if you're interviewing in industry, I think it's even more stressful because you have whole day interviews and it's extremely exhausting. And to be able to present yourself in the best way possible until the last round becomes challenging. And then sometimes you have something else after that, right? Like you might have dinner. And so it's like you're interviewing the whole day. And that's something I found. So when I interviewed with different companies, there are in some environments, interviewing environments, I felt a lot more comfortable than I did in others. And to me, that was a reflection of also how the employees were feeling within the company and the vibe they had and what kind of I absorbed from that environment. So if I felt like the environment was already tense, I mean, I was nervous. I mean, bottom line, I was nervous going into any of these interviews, but like they were also tense, then that like made it even worse for me. And by the end of the day, I was so exhausted. I remember with like one place, whereas the other place I went interviewed with, I could see people were so relaxed in their environments. And even during lunch, it was an interview lunch, and there were a lot of people who ended up joining. But then there were so many conversations about science at that table, even like within themselves. And to me, that was a reflection of how the company works inside, even when like there's no interview. And even until the end of the day, I was just energized by my interactions in the company. And I was like, this is the kind of company I would work for, as opposed to the other one where I felt very drained by the end of the day, just with my interactions. So that's also, yeah.
I think you make an absolute great point. And I think that's something that people fail to kind of not understand, but to remind themselves, you are interviewing the company just not as much as the company is interviewing you. If your gut tells you run because they were stressed and that stressed you even more for that one day, imagine 5 days a week over a couple of years. That's not sustainable. And sometimes you don't have a choice because sometimes that's the only interview you have, and you have bills to pay. We all do. But if you have the opportunity to go and interview at different places, you will always feel stressed out about it because obviously you want to show your best self and you want to make sure that they like you, but you have to like them as well.
Yeah, 100% agree. And that was really eye-opening for me to be able to gather that kind of information from just being there. And it gave me a sense of like you can actually figure out how the company works, how the team dynamics are within the team you're interviewing by just observing those kind of things.
One other thing I did with my interviewing partner or transition to industry partner is we also thought about what are some of the questions you can ask when you're having an interview with anyone, whether it's a phone call, a Zoom interview, or on-site interview. Instead of asking what's the company culture like, there's other subtle ways, like you can just Google these, to ask questions to get a sense of what's the culture like, how does the company work, like what's the dynamic, in that sense. And that also gives you subtle clues as to, is this a good fit or not?
Yeah, absolutely. I think it's so important to use the time they give you to ask them good questions and definitely prepare for that. I've been in a position to interview people since I moved out of since I switched to industry. I've had the opportunity to interview several people, and you can tell the preparation candidates have had for that question when you ask that question. Some of them are like a more generic question. Some of them are very good questions, and it makes like there'll be questions that I would not have expected, and and I have to think a little bit to answer those questions. And I think I feel like they're asking really good questions to understand, yeah, like you said, the company culture, how things work, things like how's the workload. You can ask those things in different ways, and you're absolutely right. I think you can also, as an interviewing candidate, make a really good impression if you have good questions for that question from the interviewer.
Yeah.
If you have some good questions, if you prepared, you've done your homework, yeah.
No, I agree. I think doing your homework is essential. I've been on the hiring end of things, and sometimes the worst case scenario is when you ask the candidate, "Do you have any questions for me?" and they say no. That to me is a red flag. That means you did not do your homework. You have zero interest. You just kind of applied, and it happened so that you got an interview. And also the type of questions that you get from an interviewee also matter. Yeah, I think it's important to be able to try, and but at the same time, what I was also wanting to kind of communicate here is that at the same time, you don't have a lot of time to spend with the candidate, so you have to use your time wisely, and that goes both ways. That's why doing your homework, being prepared, even for the interviewer, is very important.
Yeah, absolutely. My personal experience being an interviewee is basically I would get an agenda ahead of time of who I was interviewing with for different rounds. So the way I would prepare, I would have specific questions for every round, and if I was able to, this is what I would do. I would look up people's backgrounds and try to maybe ask them questions like, "Oh, I read this paper or came across this paper that you published," and I would have specific questions about them that shows the company that you're invested and you really care and you would really like to get this job. And I'm vice versa. I've also been impressed by candidates who did their homework on like my profile when I was interviewing them, and I was like, okay, that shows me something. And like for different rounds, I would, yeah, like I said, I would have my set of questions, and when they ask me for what questions you have, I usually have a list. I like pull out my list and like, okay, these are the questions, and we get through how many ever you can, but. Yeah, those were genuine questions I had also, but I also definitely looked up online to learn about what would be good questions to ask, and also talk to people who've done this a lot before to get their like input on what are good questions we can ask to learn more about the company.
Yeah, and these questions don't have to be, always science oriented. For example, on my LinkedIn profile, it says that I'm a cold process soap artist. And if you look at my LinkedIn profile, you can come back and say, "Hey, I saw this. How did you get into that?" And that to me is a plus, because that means that you actually did your homework. And we can have not only a very serious work related conversation, but we can have an outside work type of conversation as well about, okay, I want to learn more about you, who you are as a human.
100%. I've actually noticed that on your profile. Do you still make soaps?
I do, but I don't make cold process soaps anymore. I actually make hot process liquid soap, just because of the children. The cold process, I mean, the soap bar to wash your hands as a kid is difficult, so I make liquid soap, and I have a dispenser that goes with it so that they don't use it, and I find soap everywhere. So, but I do make it, and I also measure the pH to make sure that that I have pH paper at home. I'm being a little chemist every time I start doing those.
That's amazing that you continue to do it and you've adapted it to your current situation. And yeah, like being the little chemist, having a little chemist, like the pH testing, that's really cool too. And yeah,
Thank you. I don't have a pH meter. I just have pH paper.
Okay, but still.
I think that would be pushing it to the extreme to have a pH meter and having to calibrate it at home and measuring the pH. Although I have to say the pH paper, whenever I look at the color on the packaging, I'm like, okay, now I have to kind of guess, and I guess it's that color that I'm looking for. But I really do like to make soap.
That's incredible. I think it's such a unique, cool hobby. And it's also, I'd imagine, like good as gifts and stuff. So I always
yes. The soap bars were actually my go-to gift, and until I was making them, I had the boxes as well, labels designed. There was a website as well.
Oh wow!
So you'd get a box of 6 soaps, each different types, and so the recipes, I found some recipes online, but I adapted them. So I played around a little bit. And so you had your unscented soaps, you had your lavender soap, you had your, coffee scrub soap and stuff like that. I had a lot of fun doing it. I don't do it anymore because I don't need to. I think the liquid soap is just perfect. But if at any time the need arises, then I will get back to it. I have my little Excel spreadsheets with the quantities. Down to the well, either you're a scientist or not.
I agree. Yeah, your friends are very lucky, people who could get those soaps. Just talking about them got me into this relaxed spa mode.
Wonderful. Let's switch gears a little bit, and I would love to hear about some of the aha moments that you had as a scientist that shaped your trajectory.
Yeah, so I think we've kind of talked about this already, but I will mention it. I think earlier on in my career, when I was right out of high school, I was in a position to choose my major for undergrad. And as I was mentioning, I was interested in biology, and I was also interested in computers. And I didn't know of the existence of a field which would have both of those things. So I was in this big hall where students are basically waiting to choose their majors. And at the university where I eventually went for my undergrad, and there was a pamphlet from the university with all the available programs that they had. I was aware of biotechnology, and then I came across bioinformatics, and I was like, what's bioinformatics? And then I read it, and not a lot of people were actually picking bioinformatics at the time. People were going for biotech more. But when I read about bioinformatics, I was like, wow, this is it. Like, I even remember where I was standing in the hall. Like, I still have that memory. And so I was like, this is what I want to do. And that was done. That was probably like my first aha moment that I remember. And then another one is probably when in my postdoc lab working with GPCRs, as I said, the lab had a software which we were using to quantify allosteric communication. And there was this one evening in the lab. It was a Friday evening. I also remember that.
It is. Of course it is. It's always a Friday evening when you're alone.
Yeah, I was alone. It was like 7 p.m. It was not. Yeah, it was past. Most people had gone home, but it was a problem I was working on, is to try to quantify ligand bias. And I was just thinking, kind of trying to think intuitively, like what would make sense. And I tried to. The way I came up with it was, let me calculate the number of different communication pathways to like the G protein interface and the number of pathways to the arrestin interface, and compare that between these different sets of biased ligands and see if that makes sense. And it actually, the numbers I was calculating was starting to reflect the bias we were seeing. So that was definitely like a very important moment, because I still wasn't sure if that was good enough, like what I had discovered, I guess, until the following week. I started sharing this with my mentor, my PI, Vaidehi, and then someone else who was like a senior member in the lab. He was one of the people who developed the software. So when I shared it with him and got some feedback, they were like, "This is really cool, actually, and this is something we've been trying to solve. And so it's cool that you could do it." And that was the Boehringer project, and that's the publication that eventually came out. So I think, yeah, those would be a couple of my aha moments.
That is wonderful.
Yeah.
That is wonderful. And to your point, it always happens, on Friday night when nobody's around and you finally get something and you're like, I'm happy. Can I share this with anyone? And you have
Yeah.
to wait. And at the time, at least during my time as a postdoc and as a PhD student, there's no such thing as Slack or Google Teams where you can just post it and you say, guys, look at this. This is wonderful.
Yeah, exactly. Yeah, we didn't have anything, for that kind of I definitely didn't share anything like to the broader lab until later on.
Monday.
Yeah.
Wonderful. All right, last two questions. I forgot to ask you in the beginning. What is your favorite GPCR that you like to work on best?
This is a hard question for me. I don't have one favorite GPCR. I think it just changes with whatever I'm studying and understanding at the time. And that has evolved during my postdoc and since then as well. Because I get introduced to so many cool types of GPCRs. Like I learned about addition GPCRs way after my postdoc. And I was like, oh, I didn't know about them, but it's so cool that they do the thing that they do. It's amazing. And I didn't get to explore olfactory receptors during my postdoc, but I've learned a little more about them and how challenging they can be. And it's a whole other it's a wealth of information there that is yet to be discovered. I work with peptides as drug modalities currently. So I'm definitely looking also, I'm interested more in learning more about GPCRs in that aspect, like what kind of GPCRs bind peptides. I already had some background in this during my postdoc, working with some receptors like that, but it's a new thing. I feel like I get introduced to new types of GPCRs doing cool things all the time, and I'm more and more impressed by this family of receptors, and just think it's such a cool field to be in, to make a real impact in for human health.
Wonderful. I love it. Yeah, I think you don't necessarily have to have a favorite GPCR in order to love GPCRs and to be in the field. So definitely understanding that it's difficult to pick one among the others because they're all unique. Although some of them are grouped into the same family and they look alike, but we know that their function is definitely unique and very specific.
Yeah, I completely agree. Yeah, it's hard for me to pick a favorite. And I've also not worked with, maybe if I work with one GPCR throughout the course of my postdoc and studied that GPCR in depth, maybe that might have been my favorite GPCR, but I think my postdoc was geared more towards developing methods to study GPCRs. So I always studied a set of GPCRs and yeah.
Wonderful. Last question, toughest question of them all. What do you think we should title the episode?
Yeah, I've been thinking about that. I think it should be something along the lines of like what I was talking about is when I started working with GPCRs, it was more like, I don't know, serendipity or like a happy accident where I didn't know I was going to be introduced to this wonderful world of receptors. That's really shaped my career in a sense. I think your knowledge of GPCRs is so valuable in general. And so it should be something along those lines about how the impact they've had on my even my career, and how it was not really like a planned, it was unexpected, but it was perfect.
I love it. I love it. I love it. All right, I'll think about it. We're, as I mentioned before we hit record, we're also using a new tool that allows us to summarize these. So we'll take into consideration what you just mentioned, then we're going to come up with a title.
Okay, that sounds great.
Anita, thank you so much for your time. I really enjoyed our conversation.
Thank you so much, Amina. It's a privilege and honor to be here, to be on this podcast. And I've been looking forward to having this conversation, and I really enjoyed my time with you. Thank you.
Thank you so much. Don't go anywhere. I'm going to stop recording.
Thank you to Dr. Niveda for this fantastic conversation. This conversation explored computation as a tool to decode receptor signaling. From simulations to drug discovery, the implications extend beyond one receptor family. And they remind us that dynamics often hold the real mechanism. If you like this conversation, share it with a colleague. Subscribe to the Dr. GPCR podcast so that you never miss a conversation. And also leave us a 5-star review. It helps more scientists find the show. Thank you to the Dr. GPCR production team. All relevant links are in the show notes. And because receptors don't work alone, neither should you. Until next time, stay safe.